Hi, I'm Tammy Kielian at the University of Nebraska Medical Center, and I'm here to kind of give an overview of our manuscript that was recently published in JCI on the topic of staphylococcal biofilm and its preferential interactions and programming of granulocytic MDSCs. So Staph aureus is a common cause of biofilm infection, and in the context of this paper, we're looking at prosthetic joint infection. And a biofilm is very difficult to treat. It's caused by a collection of bacteria that coalesce into a heterogeneous community of organisms. And as such, we've shown in prior work from our group that this actually polarizes the immune response to an anti-inflammatory state and therefore prevents clearance of the bacteria. So granulocytic MDSCs are the cell type we focused on in this paper. We know that they represent the abundant infiltrate in this model, and as their name implies, they are associated with anti-inflammatory activity. So this is a really multidisciplinary effort, working with orthopedic surgeons, pharmaceutical people working with nanoparticle formulations, and people from our group to elucidate their importance in their metabolism. So we're going to be starting off with hearing from Dr. Kevin Garvin, who's the chair of orthopedic surgery at UNMC, to talk about the clinical impact of prosthetic joint infection in the patient population. Then we're going to hear from Dr. Chris Horn, one of the first authors on this paper, who's going to talk more about the bioinformatic approaches that we use to elucidate metabolic tendencies. Then we're going to hear from Prabhakar Arumugam, who's going to be talking about some of the interconnections between the metabolic pathways that we discovered. And then I will follow up at the end with an overview summary.
I'm Kevin Garvin, Chair of the Department of Orthopedic Surgery at the University of Nebraska Medical Center. I'm here today to talk about prosthetic joint infection, which is one of the most devastating complications for patients following arthroplasty. Further, this complication is associated with significant morbidity and mortality, reported to be 5 times higher than the mortality associated with a primary total joint arthroplasty. And we're often asked, why should there be such high morbidity and mortality? And I think one of the reasons is because of the biofilm associated infection with severe morbidity, as it requires removal of the prosthesis where this biofilm resides before we can cure the infection. This complex biofilm harbors the bacterial community and also skews the immune response toward an anti-inflammatory phenotype that supports the bacterial survival. Our lead researcher, Dr. Tammy Kielian, has thoroughly evaluated these biofilm characteristics and developed exciting mechanisms of cell-targeted metabolic reprogramming that could prove beneficial for biofilm treatment in combination with antibiotics. This possibility would obviate the need for removal of these prostheses that harbor the biofilm, as they could be treated in situ.
Hello, my name is Christopher Horn, and I'm one of the lead authors of this study. Prosthetic joint infections present a unique challenge due to the inherent heterogeneity of the infectious milieu. To circumvent this, we employed single-cell RNA sequencing to analyze infected tissue samples taken from a mouse model of infection. This approach revealed significant enrichment of glycolytic gene signatures within G-MDSCs compared to PMNs. Furthermore, we detected a strong signal from the HIF1a transcription factor pathway in G-MDSCs, suggesting a potential driver of the observed metabolic shift. To validate this, we followed up with a granulocyte-specific knockout of HIF1a, which resulted in a dramatic downregulation of glycolytic genes within G-MDSCs, accompanied by an upregulation of pro-inflammatory gene expression. To expand on our metabolic findings, we leveraged a novel algorithm, COMPASS, which enables the study of cellular metabolism through the integration of single-cell RNA sequencing and metabolic modeling. This allowed for the construction of in silico models of GMDSC and PMN metabolism using their respective gene expression profiles as constraints. Consistent with our earlier findings, the models revealed increased glycolytic metabolism within GMDSCs relative to PMNs. Additionally, we identified positive correlations between the increased activity of specific glycolytic reactions and the expression of G-MDSC marker genes, while the opposite held true for PMN markers. Finally, because of our proximity to a major hospital, we were able to acquire infected tissue samples from patients. However, due to the variable nature of working with such samples, the causative pathogen wasn't necessarily identical in each case, which introduced a degree of heterogeneity within our data. Despite this, we were still able to show strong conservation of the hypoxic response and HIF1 alpha activity in gene expression data, which highlights the generalizability of our findings.
Hi, I am Prabhakar Arumugam, postdoctoral fellow at University of Nebraska Medical Center. Our single cell RNA sequencing revealed that G-MDSCs have higher glycolysis and hypoxic response during Staphylococcus aureus infection in our prosthetic joint infection model. So then we asked how these metabolic pathways affect the immune function of these G-MDSCs. As we all know, glycolysis is a very crucial metabolic pathway for utilizing glucose to generate energy, which is required for basic metabolic activities and generating energy, which are again essential for the cells to function. And the byproducts from these glycolytic pathways further feed into pentose phosphate pathway and dry carboxylic acid cycle to generate molecules required for amino acids and nucleotides. So given this information, we asked how these metabolic pathways again impact G-MDSC's immune function. And in many pathological conditions, glycolysis has been shown to altered in many immune cells, and which is required for their essential function. And G-MDSCs, in the case of G-MDSCs, reactive oxygen species are shown to be essential for their immunosuppressive activity. In our study here, we have shown that using a glycolysis inhibitor, 2-deoxyglucose, glycolysis is critical for reactive oxygen species production during Staphylococcus aureus biofilm co-culture conditions. And also we have shown with hypoxic inducible factor inhibitor called ketamine that HIF1 alpha also are essential for reactive oxygen species production during this infection condition. This information leads us to ask, can we target glycolysis in G-MDSCs to mitigate their anti-inflammatory activity?
So now that you've heard about the clinical significance of these infections, the bioinformatic approaches that we took to kind of elucidate key pathways, and some of the in vitro findings that we saw with these G-MDSCs, we then moved to our in vivo model of prosthetic joint infection in the mouse. And what we did first was to use a novel nanoparticle formulation to deliver an inhibitor of glycolysis to the site of infection. This is 2-deoxyglucose. The idea is that if these G-MDSCs are really heavily utilizing glycolysis to inhibit the immune response, if we relieve that inhibition, then we should see less infection. And that's actually what we saw, where bacterial burdens were significantly reduced when glycolysis was attenuated with these nanoparticle formulations. We then went on, because we identified an important role for hypoxia-inducible factor 1 based on our bioinformatic approaches. We then went on to create conditional knockout mice where the HIF1 alpha gene was deleted in granulocyte populations. And again, this was important because HIF1 alpha can feed back to augment glycolysis. So this could potentially be a positive feedback loop. So in animals where these granulocytes, including the G-MDSCs, lacked HIF1 alpha, we saw a similar phenotype where the infectious burden was less prevalent. So this was a second independent method to really drive home the point that these granulocytic MDSCs are really relying heavily on glycolysis to organize the anti-inflammatory milieu associated with prosthetic joint infection. Another interesting observation was with both approaches, attenuating glycolysis with 2-deoxyglucose, as well as these HIF1 conditional knockout mice, we found that the granulocytes actually transformed to a more pro-inflammatory state and were harboring less of their prior anti-inflammatory tendencies. So we really did shift the metabolic set point of these cells and their functional attributes. So in summary, we think that this could really be a jumping off point for further studies to potentially leverage the information that we've obtained for potential translational applicability in the future. Thank you.